Introduce our final speaker for the morning session, Dr. Luigi Calzolai. He is from the Joint Research Center of the European Commission.
So, hello, good morning. And I will change a little bit tack because, let's say, the sizing of nanoparticle has been covered quite well in the first two talks. First, a bit about my institution. I work at the Joint Research Center, European Commission. We are Directorate General in the same function of the DG RTD that financed the Horizon 2020 program, but we are the in-house science and knowledge service for the European Commission. We provide scientific advice and support to policymakers at the European Union level. So I will talk a bit about characterization of targeted nanomedicine. So we have seen yesterday already, there is a specific session today about targeted nanomedicine. And here is just a cartoon of something that we have, a model that we have used in-house. And the new system that will be much more complex than the existing one, they're already coming into. So you have a core, either a gold or PLGA core, like here. You want a corona or PEG to reduce the interaction of this system with the protein or the plasma, blood. You want a targeting molecule, like here in this example is a polypeptide I will show you later, or it could be a monoclonal antibody or a single-chain antibody, as we heard yesterday. And maybe you want two different types of drugs, like here. So to build this, you will need definitely functional groups on the external surface. And these products are difficult to produce. They are difficult to characterize and to evaluate. So they pose problems both to the people that want to develop this, to the pharma companies that want to bring on in the clinical trials, but also to regulators. And there are difficult regulatory assessment of this innovative medicine. And I will show, and this is basically covers the physical-chemical characterization and what Jeff described before in the second talk. So you can start from simple characteristics of your system in terms of size and charge. You want to go on maybe with particle size distribution with more sophisticated methods. And here, I'm not going to cover, but basically we use a lot. Our workhorse is basically the fourthly flow fractionation with DLS and MALS online. You want to go proteins bound to nanoparticles. And then, if you have an active targeting, you want to know the structure and the integrity of your targeting molecule. And I will give you some example how we address this in our work. So these are a multifunctional gold nanoparticle. We recently published this on nanoscale. And this is our model system where we built two different functionalities on the particles using a tile PLOH and tile PLCOH. And so we arrive at the gold nanoparticle where you have, ideally, you hope you have low binding of plasma protein thanks to these repeats of this group, and then to the functional groups with additional chemistry where we can go on. On saturation, we arrive around 3,000 PO molecules per nanoparticles, and going on with the standard activation chemistry, EDC and HS, we can bind a protein. In this case, we use, as a model, human cell albumin. So the problem is, I give you this presentation, I call this PowerPoint synthesis. How do you know that I really have 3,000 PO molecules per nanoparticle, or they really bind these proteins on your nanoparticle? This is the method that we are using. It's not the only one. We have developed this in the last few years. And so basically, it's a combination of measurement using FFF-DLS that gives me the accurate size of my complex. Here, because it separates based on size. And then we measure exactly the same sample with this technique, the centrifugal particle sedimentation, where the data, the time that the system needs to arrive to the detectors depends on the density and the size of your sample. If you combine the two information, and we get the density, and then we get the mass of the functional layers, and we can sort of estimate, I would say, the number of ligands per nanoparticle. So what is the effect of ligand density on the properties of this system? If we start with a 10% of pOH on the total possible surface coverage, we estimate around 570 per molecule, or we can start from 100%. And these two systems, they have very different properties when you incubate with human plasma. Here is that the two most common proteins in human plasma is human serum albumin and immunoglobulin. And you will see that when you have a 10% coverage around, you still see some binding of the proteins to the nanoparticles, while when you have complete coverage, we don't see any binding of proteins. And how are you going to measure this? There are several ways. Well, not that many at this moment, but we went back very, very simple, very basic in a sense. This is SDS page. This is the standard technique. I used to produce my own recombinant proteins. And here you have the size, the different size that you can get in human serum. If you incubate gold nanoparticles, citrate stabilized, so let's say bare, non-functionalized, you see it binds a lot of different proteins. When you go at 10% pale coverage, you still see some binding of mainly HSA, but also some other proteins. When you arrive here, 100% coverage, you eliminate probably more than 99% of the protein binding. So with this system, we can also move on. And again, we had developed this in the last years. You start with your system, it is a standard activation chemistry, separation of the complex with the FFF, and then we can measure the structure of the protein using circular dichroism. Either we measure with our benchtop instrument in our lab, or because we have worked with very low concentration, we can go to the circular dichroism beam line at the Diamond Light Source. This is a synchrotron radiation in Oxfordshire in UK. And using circular dichroism, what you get the secondary structure elements on your proteins. Without too much into the details, you can sort of, if you say the free HSA, it's an alpha helical protein with 72% alpha helical elements and a little bit of unordered structure. And if you see here, the analysis for the HSA bound, chemically bound to the COH group here, you have a very similar amount of secondary structure. So you know that your secondary structure is conserved. I don't have the time here, but actually you can go on. You can do more than this. You can measure the stability of the protein in the different sample using different type of experiment with circular dichroism. The other example, again, this is our internal development using targeted PLGA-PEG nanoparticles, where in this case we attach a substance-P. It's a polypeptide 11 amino acid that binds the neurokinin 1 receptor that is overexpressed in malignant gliomas. Similar way, COH group activation, and going back to very basic NMR, just 1D NMR, very low field, you can see very easily follow the attachment of your polypeptide to the particles. But then you can do something a little bit more sophisticated using, again, this is really basic, 2D NMRs. You can check at the atomic level control of the binding. So in theory, you expect that the activated particles will bind to the primary amine present in the system. But here, normally you think that will bind to a lysine, but there is also the NH2 terminal group. And actually, our data, our evidence tells me that probably it binds here rather than here. But apart from this is a technique where you can really follow where the binding of your targeting molecule happens, and you have a very tight control at the atomic level. And there are not many techniques that can give you this type of information. So just going back, there exists infrastructure in EU where you can access this type of expertise and some measurements. So the European Union Nanomedicine Characterization Laboratory that Adriele was mentioning before, and there is a specific session on this tomorrow afternoon. And we have also our we can access a little bit of our expertise via this Open Lab program at the JRC, where you have access to very small amount of time for measuring something very specific. Finally, so in conclusion, if you combine FFF DLS with, so let's say, accurate measurement of size, in our case, centrifugal particle sedimentation, you can, well, definitely get the accurate particle size distribution of this very complex object. And you have a way to quantify the number of ligands per nanoparticles in terms of functional groups, proteins, or polypeptides. You can do more sophisticated, basically using a biophysical technique that exists since a long time, and definitely by circular dichroism, you can check the structure, at least the secondary structure elements, of your proteins when it's bound to a nanoparticle. Using NMR with smaller peptides, you have an atomic level control of the binding. And then you have access to infrastructure and expertise available in EU, especially the UNCL characterization of nanomedicine, where all this work will be done for free for companies, groups, spin-off, they go through the process. Let me acknowledge my people at JRC. We are a group of people we work together since a long time. And Robin is, really. The postdoc that did, Robin and Isa did most of this work. The people at the CD beamline facility at the synchrotron, like Sir Giuliano Siligardi and Rohane Hossein. The whole UNCL team. And finally, thank you for your attention.
Any questions?
Yep.
With the ligand binding, when you're measuring the size of a particle, say with 500 ligands versus 3,000, I mean, isn't the diameter essentially the same? And wouldn't it be easier to use charge? Electrophoretic mobility changes as you put more charged ligands on, because they had a carboxyl group on the end.
So here I had only 15 minutes, so I had to choose the data. So if you go, I can give you our original article. We follow this using charge, using size, using different methods. But we do see also a small change in size, if I remember correct, but I have to go back to the original data when you see a partial binding, let's say saturation at 10% instead of 100%. But definitely we follow this also with a charge, and we see a difference, yes.
Any other questions?
Is it available to know the uniformity of ligand on the surface draw FFFDLS? You know, 100% of ligand on the surface, it's not mean that all the surface is covered, okay? It can be half of the surface and the half is nicked.
Yeah, that's, so basically this can happen. I don't think we have a simple way to check this. But what we have done here, again, I had to choose the data. We did a sort of titration in a sense that I show only 10%, 100%. Actually, we went 10, 50, 60, 700, 5x, 10x. And so we see a sort of saturation behavior. Well, we do really see a saturation behavior. And then we compare this with the theoretical amount that you can put on a surface, on a given surface of 15 nanometer sphere. And those two numbers, they match pretty well. So in this case, we are sort of confident that what we see, it's complete coverage, and probably they are sticking out as you expect. And these are they are self-assembled monolayer-derived type of ligands. So in this case, I'm quite confident. When you go to more complex or different systems, I'm not completely sure, obviously. You have always to check. In terms of the FFF, the good part with the FFF in combination with DLS and MALS is that you don't rely just on the exit time. So with the FFF, obviously you can size based on the exit time and the calibration. But when you put online DLS and MALS, you measure the real size over the exit. So even if you have difference, and actually can give you a lot of information, you can have differences a little bit in the chemistry, on the external surface, but still you get the right size. And actually, it's a very good way to control, even check batch-to-batch comparabilities. Yeah. So I would say that platform that Jeff was describing is really powerful. We have example of this.
Any other questions? Okay, so before we begin the next portion, which is the questions debate, I want to first thank all the speakers.